Okay, so there are 3 types of monohybrid crosses that'd be really helpful in us understanding how to read data based on genetics problems and genetics crosses. So you could always pause this video at the end and write these down. I think that if you're a student studying genetics, that would be helpful and recommended. So anytime you have a cross that includes a parent that is true breeding for a dominant trait, because this represents a gamete, the Mendel's law of segregation, 100% of this individual's gametes will have this dominant allele. Therefore, it only has dominant alleles to offer its offspring. Therefore, 100% of the offspring will have the dominant phenotype. And in this case, if it's crossed with a homozygous recessive, then the genotype will also be the same. But in reality, it doesn't matter what you cross it with. You could change this and it would still have the same phenotype. It all comes down to the fact that one parent is true breeding for the dominant trait, and therefore the only possibility in the offspring is to look dominant and have a dominant phenotype. Now the next one is if you have a heterozygous crossed with a homozygous recessive. You can see here that this parent in half of their gametes will have a dominant allele, half their gametes will have a recessive allele, whereas the homozygous recessive parent only has recessive gametes to offer. So when we look at the probability in their offspring, we see that every time they have an offspring, that there's a 50-50 chance or a 1-to-1 ratio that they will have the dominant or the recessive phenotype. For every one dominant phenotype, there is one recessive phenotype born. And in this example, the genotypic ratio is also 1-to-1. Now, when we look at a hybrid or a heterozygote crossed with another heterozygote, we find that we get a fun-looking phenotypic ratio. One of their 4 offspring will be homozygous dominant. 2 of their offspring out of the 4 will have a hybrid, and then 1 will be homozygous recessive. Now, this is not saying that if they have 4 children, it has to look like this. What this means is that there's a 25% chance every time they have a child that it'll have the homozygous dominant genotype. If they have, every time they have a child, there's a 50% chance, 2 out of 4, that they will have a hybrid as an offspring, and then a 25% chance, 1 out of 4, that every time they get pregnant, the child will be homozygous recessive. So when we look at the phenotypic ratio, what we see here is that 3 out of 4 likelihoods are dominant phenotypes. So there's a 75% chance or a 3 out of 4 chance that every time they get pregnant or have an offspring, the baby will have the dominant phenotype. Could they have one kid and the first time they ever have a baby, it has a recessive phenotype? Sure. There's a 25% chance of that happening. How about the next baby? Sure. 25% chance of that happening every time they have an offspring. Now the genotypic ratio here is what's fun. When you look at this, you see a 1-to-2-to-1 ratio. 1 homozygous dominant, 2 hybrids, and 1 homozygous recessive. So let's go ahead and practice with this a little bit. So in a population of giraffes, a true breeding tall giraffes are crossed with true breeding short giraffes. All of the F1 are tall. What are the genotypes of the parents and the offspring? So here, it doesn't directly tell us what's true or what's recessive. It just tells us that we have a true breeding crossed with a true breeding. So we don't know if tall at this point is homozygous dominant or homozygous recessive. And so we look at the F1 though, and in the F1 we see that they are all tall. That means if you had a true breeding parent here and a true breeding parent here and they were crossed and you do the Punnett square, all of the offspring are going to be heterozygous. And therefore we know because they're heterozygotes and they exhibit the tall phenotype, that's how we can narrow it down that tall is the dominant allele. So there you go. Now in a different area, scientists made a mistake and have a large F1 population but don't know who the parents are. There are 86 tall giraffes and 27 short giraffes. What are the genotypes of the parents? So you want to question the ratios that we saw back in this slides here. You want to think about those phenotypic ratios. And what we see is that the 86 to 27 is closest to like 90 and 30, or like a 3-to-1 ratio. So then your next step is, okay, well, I have my ratio. What combination of parents lead to that ratio of offspring? and you find, oh, it's when I cross a heterozygote and a heterozygote, I get a 3-to-1 ratio. Okay, so if there are 37 tall giraffes and 43 short giraffes, what are the genotypes of the parents? Well, here I see that my ratio, my data is about a 1-to-1, and I know that is when I cross a heterozygous with a homozygous recessive, I get a 1-to-1 ratio. And if there's 233 tall giraffes and 0 short giraffes, what are the genotypes of the parents? In this example, I know that at least one parent is homozygous dominant because every offspring inherited a dominant allele. And our last question here, in a population of leafcutter ants, 1,837 are born with a brown body, 598 are born with a gray body. Which trait is dominant, which is recessive, and what are the genotypes of the parents? So take a minute and think. And here we have that this ratio is about 1,800 to 600, which is a 3-to-1 ratio. So we know that the parents are both hybrids, with the majority of their offspring having the brown phenotype. So brown would be the dominant allele, and gray would be the recessive. So hopefully this little introduction on understanding data and genetics was helpful.